Hi Year 11, welcome to the first of 4 Biology Paper 1 mind map lessons where we're going to talk about cells today. I'm going to talk you through the key spec points as I go through the mind map. Feel free to just listen along or you could create your own mind map and kind of pause throughout. It's completely up to you. I will do both foundation and higher content. That'll make it clear the bits which are kind of just relevant to people doing the higher paper. So if we start off then with cells, the basic kind of building blocks that build up all living things. Looking at cell structure, there are two types of cells that we need to know about for the GCSE course, and those can be broken down into prokaryotic cells, which are the ones that contain no nucleus, and instead tend to have either like a single DNA loop or a plasmid, and those are your typical bacterial cells. Okay, so prokaryotic, no nucleus, for example, bacteria. The other type are the eukaryotic cells, which do contain a nucleus and membrane-bound organelles, and those, for example, would be your plant and your animal cells. So, thinking then about your prokaryotic cells, they contain key subcellular structures such as cytoplasm, cell wall, cell membrane, DNA loop, and plasmids, which are also circular DNA. With your eukaryotic cells, we need to bear in mind the things that are included in both animal and plant cells, and also the ones that are just in plant cells only. So, if we start off then with the both section, so plants and animal cells, we've got things such as the cell membrane, which controls what enters and leaves the cell, the cytoplasm, which is the jelly-like substance where all the chemical reactions take place, the nucleus, which controls the cell and contains all of that genetic material, the DNA, and ribosomes, which are the site of protein synthesis, so making proteins. And then the last key subcellular structure for both is the mitochondria, which are the site of aerobic respiration. Okay, so if we're then just looking at plants only, the three main things that they contain that are not found in animal cells are the vacuole, which contains cell sap and provides like a support structure to the plant cell, the cell wall, made of cellulose, which also supports the plant cell, and the chloroplast, which are the site of photosynthesis, and they contain that green chlorophyll pigment. When it comes to labelling or drawing plant and animal cells, the key thing to remember when it comes to sort of the cell wall and cell membrane aspect is the fact that when you're drawing a plant cell, the cell membrane is the inner kind of envelope structure, and the cell wall will be the outer one of the two. Okay, so if we then move on to start to look at stuff kind of relevant to microscopes, the key thing with this, first of all, and it ties into sort of GCSE maths at this point, is the idea of scale and size. So we need to be familiar with being able to convert between our different measurements. So we should hopefully know that a metre contains 100 centimetres, therefore to go from a metre to centimetres you can multiply by 100. To go from centimetres to millimetres, you need to multiply by 10, because there's 10 millimetres in every centimetre. To go from millimetres to micrometres, it's multiplied by 1,000, and from micrometres to nanometres, it's multiplied by 1,000 as well. Obviously, if you're going the other way, you divide by the same value, so you can see that on the diagram there. The key one really to bear in mind for this, which tends to come up time and time again, is millimetres to micrometres. So it's just important to remember 1 millimetre, contains 1,000 micrometres. So that bit there that I've highlighted is really, really important. If you're only going to learn one of these, that is the one to definitely memorise, though hopefully some of the ones at this side we should already know. Thinking then about microscopes, the key thing to remember with that is the I-AM triangle. So I representing the image size, so that's referring to what you're actually seeing through the eyepiece lens. A is the actual size, so the physical size of that thing that you are observing under the microscope, and M is the magnification, which is how many times larger the image is than the real object. Okay, and that's the one that usually obviously is, 100 times or something like that. So there are two types of microscope that we need to be aware of. The ones that we've used in class will be light microscopes, so they're good for looking at cells, and particularly ones that are kind of quite sizable. Electron microscopes, however, are much more expensive. They're much more powerful in that they have a better resolution. So these are the type that only really universities tend to have because they're really expensive bits of kit. And by resolution, what we're referring to is the ability to see two or more objects as separate objects. And so that's just worth bearing in mind with electron microscopes. They're much more powerful because they have a much better resolution. Okay, moving down here then to have a look at transport in and out of cells. We've got our three key transport processes that we need to know really well. So if we start off with probably the easiest one to understand, which is diffusion. Diffusion, from a textbook kind of definition point of view, is referring to the movement of particles from an area of high concentration to low concentration. So that's the one where you're thinking about, for instance, if you spray a perfume or an aerosol, when it's leaving the can, it's going to be in a really high concentration. There's going to be lots of particles coming out of the can. They are going to gradually, passively, without energy being needed, disperse to areas of low concentration. Okay, so from high to low, no energy is needed for this. It's passive. This can be affected by things such as the concentration gradient. So how different is that kind of difference between the high concentration and the low concentration, the temperature, or the surface area. Okay, so that's your three factors that can affect diffusion. Moving on to osmosis then. Osmosis is referring to the movement of water molecules from a dilute solution, so where there is a high concentration of water molecules, to a concentrated solution, where there's a low concentration of water molecules, via a partially permeable membrane. Again, with osmosis, no energy is required. Moving on to active transport then, this one is the one where energy is required, and that energy in active transport comes from respiration. So with active transport then, you're talking about the movement of particles from an area of low concentration to high concentration. So it's going against what we call the concentration gradient, and it's, the opposite of diffusion. Therefore, energy is required to force those particles to go against that concentration gradient, so moving from low to high. Okay, so carrying on then, we're now going to have a little look at the cell cycle and mitosis. So if we start down here with the cell cycle in general, there's no real official kind of start or end to this because obviously it's a cycle. So what we'll do, we'll start down here for today. So if we look at this box first of all, one really important part in the cell cycle is that each cell is going to grow and it's going to make new subcellular structures. Then the DNA is going to be copied, so every chromosome is going to replicate itself. And then mitosis is the kind of third step in the cell cycle, where that division of the chromosomes, the cell membrane and the cytoplasm actually occurs, and then the daughter cells are formed. Okay, so those are your three separate stages. It happens obviously in a continuous cycle. So looking then further at actual mitosis, the whole kind of product of mitosis is two identical daughter cells, each containing 46 chromosomes. So the way in which this happens is you start off with one adult cell here containing 46 chromosomes, which is what all human body cells contain. Each chromosome replicates itself to make 92. Those 92 then split to form two identical daughter cells, each containing 46 chromosomes. Okay, and obviously in that splitting process, the cell membrane and the cytoplasm have to actually divide so that two new cells are formed. Looking at chromosomes in slightly more detail again then, it's important to remember that they're found inside the nucleus of cells. So inside the nucleus you'll find chromosomes. The chromosomes are made up of DNA, and then just to add on to that, sections of DNA are what we refer to as being genes. Body cells in humans, as I've said already, contain 46 chromosomes in total, and they can be also described as having 23 pairs because every one of those 46 matches up to a corresponding pair. The gametes, which are the sex cells, so the sperm and egg cell in humans, contain only 23 chromosomes. Okay, so 23 single chromosomes are found in those. Moving on to stem cells then. Over here. So, with regard to stem cells, we'll start at just kind of looking at where they come from. So in plants, this is hopefully nice and easy to remember because there is only one source of stem cells, and that is the meristem, which is the growing root or shoot. So the meristem tissue is where those stem cells are found. In animal cells, or in humans, there's 3 locations that they can be found. They can be found in embryos, the umbilical cord of kind of newborn babies, and the bone marrow in adults. So there's 3 locations in humans. What stem cells are then is they are undifferentiated cells. So by undifferentiated we mean they've not yet become specialised. We're going to look at specialised cells in a second. So they are undifferentiated or non-specialised cells. They can be used for various different medicinal purposes and treatment. The 2 main ones you need to know about with regard to humans are treatment of diabetes and the treatment of paralysis. So those are the 2 to remember. Following on from this then, it does link in with therapeutic cloning to an extent. Now, therapeutic cloning is the process in which clones are produced to treat diseases but will not be allowed to develop into offspring. So that's worth bearing in mind as well, because that offers another avenue for treatment of various different human diseases and things like that. So the final section then that we need to look at just to finish this cells mind map off is the specialised cells. Now, these specialised cells are ones that have undergone cell differentiation. So it's when stem cells undergo differentiation to become specialised cells, and in that process of undergoing differentiation, their shape will change, they will gain or lose different subcellular structures, and the reason that takes place is so that they can carry out their specific function. So, to give you a few examples then of key ones, in animal cells, we've got the sperm cell, egg cell, neuron, and muscle cell, all of which have different shapes, slightly different subcellular structures because they have different functions, they've got different jobs. And then in our plant cells, we've got root, hair, cell, xylem, and phloem. Okay, and we'll come to look at those in slightly more detail in some of the later topics. Okay, so that's kind of it in general for the cells aspect of things. And actually all those things we just talked through then are relevant to both higher and foundation tier at this point. So that's it for cells. Join us for part 2, which will be looking at unit 2, which is organisation. Keep up the hard work.